This episode is with Dhananjay Huilgol, a senior research associate at Duke University in the Department of Neurobiology. He is currently focusing on embryonic development. Thank you for being here, and I hope you enjoy this episode. I'm doing very well. Thank you. Thank you for joining. Thank you for making time.
Oh, thank you for inviting me. It's a great opportunity. It's fun. It's fun to talk
Yeah,
to college students. Yeah.
I completely agree. It's fun to talk about things that you think are fun in the first place.
Yeah.
How are you
So
doing today?
I'm doing very well. Thank you. How are you?
I'm doing great as
Am I
well.
audible? Uh, am I fine or?
Yeah, you look, yeah, you hear fine, you'll fine. Everything's good.
Okay, great. Okay.
Yeah, I also, everything is once I finish recording, I think I told you, I always edit everything.
Right.
And so, I'll go through the video, and if for some reason it's not loud either from you or from me, or it's too silent, I can raise the volume and I can work on
Oh,
that.
oh, okay. That's great. Okay. That's
Yeah.
fine.
So no, don't worry about it. Everything looks
Okay.
great.
Okay. Okay. Yeah. What made you think about these podcasts? Like what made you decide on doing these?
Yeah, so the main reason I like to do them, I wanted to do it, is because I like to learn, but it's not just learning in general. A lot of times in school, you get taught things that you not necessarily find very interesting, and so you kind of have to just push through it, and sometimes you don't even learn that well, just because it's something that you don't like very much. I'll give you an example, just to put it out there. I think I told you I'm majoring in business technology. It's kind of like computer science, and a lot
Yeah.
of times, as part of the course, I have to take accounting classes, and I don't like accounting, right? And so I have to learn accounting, and I don't enjoy it.
Okay. Okay.
And so with these podcasts, what's cool about it is I get to invite people to talk that I find are in interesting fields, and so I
Hmm.
enjoy having that conversation and learning. And what's cool
Right.
about it is I'm sure other people like to, there's a lot of curious people out there, so they like to learn about these things, and putting it out there, whoever wants to listen and learn, it's always a fun time.
Yeah. That's great. That's great.
Yeah, that's the main reason. Yeah.
That's nice. Yeah. It's good to find new ways of learning, and this is great. Yeah.
Yeah.
And
So
I guess you can also teach others through this.
that's yeah, that's right. Like it's not just me being able to, I guess, fill the need for my curiosity, but also having the opportunity to, whoever wants to listen and
Uh-huh.
learn about it, they can do that as well.
Yeah. Yeah.
And it's also very cool because it's um I invite a lot of people, like different backgrounds, right? And so, for example, the other day I was talking to a history professor, or earlier today, actually, I was talking to um a coach of mine. He's a mental coach, so he works on
Oh,
um
wow.
like mental training and sports psychology,
Uh-huh.
and things like that. And so it's I get to talk to all these different people, and whoever might want to listen. Each person has different interests, and
Right.
for someone, a particular topic might not be very interesting, but
Hmm.
then some other day they'll find an episode that is something that they're interested in. So it's a
Yeah.
wide variety, which I think is pretty neat.
That's nice. Yeah.
Yeah.
Cool.
So what do you do? And why don't we start with that?
Okay. What do I do? I am now a senior research associate in Josh Wang's lab. Josh Wang is a professor in neuroscience at Duke University. And I moved with him from Cold Spring Harbor Lab. I was a postdoctoral fellow with him for the past few years. It's in New York. And before that, I did my PhD in developmental neuroscience in India at the Tata Institute of Fundamental Research. I have been working on how different aspects of the brain are formed within the embryo, and what are the mechanisms, cellular and molecular mechanisms that actually tell a particular structure to become what it is. So for example, I work right now on what is known as cerebral cortex, which is the major seat of intelligence and cognition in the humans and other species, other mammals as well. Um, so I learn about what makes a neuron of the cortex that particular neuron. How is that decided? Are there different molecules within the progenitors or the mother cells that will define that this is how it will become this particular neuron versus type B neuron versus type C neuron? So that's basically, in short, this is what I do.
Right.
We work on mice as model systems, because we directly cannot have access to human tissue. And mouse has a very similar brain structure to humans, but also different in a lot of ways. So there are things that we can model, and there are things that people, scientists in the field, are trying to model in a dish from a human tissue. So, something like that.
Yeah, I know that a lot sometimes in research, some people once they die, they can donate their brain
Exactly.
to research. Do you also use brains from people that have passed away?
Yeah, so in our lab we do not so far, but I think we are planning to. And what Because I'm doing development, basically our access to certain fetal or embryonic tissue is more valuable, and we have certain members in our lab who are actually medical doctors as well, and so they have access to that kind of tissue. So we can actually see that what we are studying in mice, will it be applicable to humans or not, and so forth.
Do you study mice of any age or like in the embryonic stage?
So what we do is we manipulate the embryos, or we do experiments at embryonic stage, and then we look at them in the adult to see if whatever we did in the embryo, has it actually affected the functionality of the brain, or has it changed the kind of neuron it had to become in the adult brain, or something like that. So yeah,
Okay,
we
that's
look at
interesting.
different ages. We look at the embryo, we look at the adult, but all our experiments are done at the embryonic stage.
Okay. Now, this is a very, I want to say somewhat specific aspect of neurobiology and neuroscience. Is that always something you were interested in, or during your PhD in India, how did you get to this point?
Yeah, I that's a very interesting question, and I have a story to tell.
Okay, go
because
ahead.
uh, because I as an undergrad I was very interested in immunology, and the reason was that there were these multiple molecules and multiple cells that become a specific type to fight infections, to fight any external agent, right? And so when I joined my PhD program, I did rotations like grad students do, different 6-week rotations in different labs. And at the same time, I had a Neurobiology 101 course, which was taught by one of our professors in India. And I really liked neuroscience. And at the same time, I was doing rotation in a lab that was developmental neuroscience lab. And it was very fascinating to see a neuron grow inside in a dish, and we had like fluorescent proteins that were expressed in a particular cell, and you could see axons, which are basically processes that move out of the neuron to target different other neurons. I could see it grow, and so that was very fascinating. So turned out that the mechanisms, different molecules that play a role in making immunological things like T cells and B cells that actually fight infections. Similar process happens when a brain is also formed, where different molecules interact with each other to tell a particular mother cell that this is the neuron you have to produce, and this is the second neuron you have to produce, and so on. So I think that drew me to neuroscience and the brain very early during my PhD. And I joined that lab in which I was rotating, Dr. Shubha Tole's lab, did my PhD, and that's how I entered the field of developmental neuroscience.
Right. So it's very similar to the immunological process of a cell.
Yeah, so basically what happens in an immune system inside the body is that different cells become different types based on the kind of immune challenges that they face. And then there is a role of molecular interplay within each cell that will tell them that this is what you have to become. So I sort of became interested in that sort of a developmental process. So how does a cell develop into something else? And that's what drew me to developmental neuroscience, because brain fascinated me, and at the same time, development fascinated me. So I was like, might as well study something that has developmental neuroscience, which will connect both my interests at that time.
Right. I agree. I think the brain is one of the more interesting things
Yeah.
when it comes to the human body, just because it's pretty much, your everything, it controls everything.
Yeah, it does.
And there's so many aspects of it that we don't know enough about, I feel, yet. You have the dreams, and then you have, what actually defines and what is consciousness, and all these
Yeah.
different things which
Yeah,
get
and
very
those things are
complicated.
far-fetched. Even as scientists, we do not understand these things, what is consciousness, and a lot of research has been done on sleep and wakefulness, but we still try to understand what dreams are, what they're constituted of.
Right.
Yeah, I like to study the fundamentals, where how the brain is built in the first place. And if that goes wrong, then the function goes wrong automatically. So
Right. Now you mentioned you had I believe you said you did your PhD in India.
that's
It,
right.
it must have been very different. For me, I went to the US, or came to the US um for my last 2 years of high school, but before I lived in Mexico, and all of my education was in Mexico.
Right.
And it's very different, the formatting and just the curriculums that we get taught. It's just very different to here. I'm sure you've experienced a lot of differences as well, and similarities. Why don't you explain a little bit of how is what's moving here and so on?
Sure, yeah. So the major similarity was, so I come from an institute named Tata Institute of Fundamental Research, where we have PIs, or principal investigators, or professors who actually do their PhDs or postdocs in the US or in other countries in Europe and so on, and then come back to India. So the kind of training I got from Shubha and Vidita, who were my major mentors. Shubha was my PhD advisor. Vidita was a thesis committee member. And because of their training in the US, I got a very similar training back in India as well. So we had a rotation system. The major difference between India and here is that we never called our professors by their first name before that. So like till undergrad, we used to call everybody ma'am and sir, all our teachers were ma'am or sir. But what happened at TIFR, which is my institute, is that we were trained to first call them by the first name basis. So that's why I call them Shubha or Vidita or something. And that itself broke the ice. It was an icebreaker. We could talk freely. We could discuss and argue freely. We had a rotation system. We really had a good time understanding what publications mean, and publications in international journals. And so even though the opportunity in India is a bit less, I was lucky enough to get admitted to this program, where it was very similar to the kind of system that exists in the US and in Europe. Uh, I think one major difference was that we do not have or rather, I did not have a full department that was dedicated to neuroscience. So we had people from different fields. So we had cell biologists, we had neuroscientists, we had people who did biophysics and so on in the same department. So our department was a broad biology lab that had the advantage of us being able to learn about different aspects of biology, and at the same time different aspects of science, because we were in an institute where there was chemistry and physics and computer science and so on. But at the same time, a lot of peer reviewing that happens here, the ability to attend conferences every year, or every 6 months, and those kind of things was something that we lacked back there. I think things are changing now, because it's because of globalization. There are more opportunities for people to come outside and attend conferences. Despite that, I did attend a Society for Neuroscience annual meeting, a Gordon Research Conference, and I went for a 2-week training to learn a technique in Japan. So I think my advisor and my department actually did a very good job of training us in terms of how international research is done. And so for my postdoc, because we are not familiar faces, we don't meet other professors in the US or in Europe on a day-to-day basis or in meetings. I had to write to them. I had to actually meet them personally. I actually I flew down to the US to interview with a bunch of labs. And finally, I shortlisted some based on our mutual interests. And then I joined here.
Right, right.
Culturally, it was very different. India is more, people are more together and they are basically in each other's face, which is a good and a bad thing. Whereas in the US, it's more individualistic lifestyle. So that took some time to get adjusted to. But
Right.
but I think in terms of resources and the access to resources, the lab I joined here, Josh Wang's lab, actually gave a whole new experience in terms of what you can do with the number of techniques you have to answer a question.
Yeah, I think there's this perception, same as with Mexico. I am from Mexico. I am from there, and there's when it comes to somewhat less developed countries than the US in terms of its economy, its technology, all these different things. I think sometimes there's this perception that when it comes to medicine or some field like neuroscience, for example, like you said, the access to resources is going to be at least somewhat different, and the availability to have the latest technology.
Yes, I think, yeah, that's exactly the point I was making.
Right.
Yeah.
Okay. So when you joined here in at Duke, those things were different.
Yeah, they were
Right.
different. Even though I think the way to think about science and how to access science and approach science was taught back in India. I learned it during my PhD. I think at my postdoc, I did learn about what are the different resources that one can access very easily. And I think that helped a lot in
Right.
terms of, if I want to go back to India, which I do intend to, I can take some of these resources back and actually develop a good system over there.
Right. It is some going back to the research that you do.
Right.
I sometimes again, an interest of mine is neuroscience, psychology, and I've looked into looking at graduate programs for those things. The thing that kind of holds me back is um like I feel like you either have to, when it comes to psychology, at least, you either have to do research once you finish those programs, or you become a therapist or a psychiatrist or something along those lines. Is there, and with neuroscience, for example, I mean, you can become a neuroscientist or and a doctor. What other opportunities, you talk about your specific research, what career opportunities are there in that area?
Yeah. So now with the coming up of a lot of biotechnology industries and a lot of startups, I think not just the academic route, which I'm talking about, but also getting into industries, making drugs that will help change something, say, if so I'll give you an example. So there's some mothers, some pregnant moms may have alcohol addiction, and that may affect their babies, and that can actually be treated with drugs now, where you can help the developmental delay and not let that affect learning ability or something like that. So, those kind of things can come from this, and it can go into industrial research in that sense. Knowing more about broad molecular biology can also help you get into forensic sciences and try and understand how molecular biology can be used for, tracking down criminals, for example, or tracking down certain agents that were used to commit a crime. Apart from that, you could become a teacher, and a lot of big data science happens in neuroscience. And so you become a data analyst. So I think you can go into data management and data analysis. And a lot of my friends have actually done that. Um, Not specifically neuroscience, but in general, the analytical skills that you acquire during a PhD helps you also become a very good consultant. So a lot of consulting companies, including BCG and McKinsey and Bain, these big companies actually look out nowadays for PhDs in general, and you can get into consulting. So I think science or just doing this kind of research can give you different avenues to get into different programs, whether it's academic research, whether it's industrial research, or getting into forensics, getting into teaching, getting into consulting. So I think there are tons of opportunities that are there, even if you're not. So if you realize very early on after your PhD that you don't feel like becoming a researcher, because it does take a lot of toll on your personal life and so on, you can change your career even after that. So, and
Right.
nowadays, I think, so at the time I did my PhD, these opportunities were not so available or talked about. But now I think a lot of grad students and a lot of undergrads actually come with this kind of training, with this kind of mindset that they want to explore different avenues and different approaches. If they cannot do a PhD, or if they do a PhD and they don't want to get into research, there are these multiple avenues you can get into.
Right. You know, yourself as a, you know, primarily focusing on research, do you have some sort of plan for the future, or do you want to focus on research right now, at least for the foreseeable future?
Yeah, so I have spent quite a long time as a PhD and as a postdoc to try and develop a research program that I would like to carry out in my own lab. And I mean, right now I am at the end of, writing papers and getting them published and so on. And once that is done, I am parallelly creating an application and a foreseeable research program for my own lab. And I'll be applying across the globe. uh, my primary goal is to get into one of the good places in India. If I cannot, then somewhere closer. And hopefully, I'll have my own lab as an assistant professor and have students and postdocs and train people like the way I was trained. So, yeah,
Okay, so you'd like to go into the teaching route?
I want to go into actually research route, but with a
Okay,
bit of teaching.
with a bit
I
of
want to
teaching, right?
have I want to have my own lab where I can do research on developmental neuroscience, but at the same time, I would like to train younger minds and teach certain courses where they can get interested in development.
Right,
Because even now, like you said, in Mexico and in places like India, neuroscience itself is small, but developmental neuroscience is even more focused. So I think not many people have that opportunity, and it would be great for people to know what these things are and what these
right.
career opportunities are. Yeah.
Yeah, it's interesting you say about all these career opportunities. I think it's something that isn't necessarily as widely available, or the information about
Yeah.
these career opportunities isn't as available, especially if you're not maybe someone that's more, in depth into that specific field. And
Yes.
so I think maybe, it's hard to find these things that, you're sharing to me right now.
So
That
there
you
are, sorry,
No,
there
no, no,
is
Go ahead, go ahead.
there is a New York Academy of Sciences, which is what I'm aware of because I came only after my PhD. So there was this NYAS that gave us a lot of opportunities, gave us a lot of exposure to these different careers. Um, Cold Spring Harbor itself has this public outreach kind of forum where we can actually talk to other people and find out what careers can exist. Oh, I forgot to say something about writing. So there is science journalism and there is medical writing, where you are a liaison between people who do clinical research versus people who are in industry, and you can actually be a writer and communicate between these two parts of the same company. So I think these opportunities also come as you do a PhD. And so New York Academy of Science, there is AAAS, which is an organization for the advancement of science, which also has the Science magazine. So these organizations actually open up a lot of career opportunities for students and postdocs.
Right.
So you can approach these. Yeah.
I think
I'll
you
be
talk.
happy to send you. I'll be happy to send you a link to these sites so that
Yeah, or
if you,
I'd like that.
yeah,
I think, the more information is always better. You can make a more
yeah,
informed decision on whatever it is you're doing.
that's correct. Yeah.
I was you mentioned something about, I believe it was, biotechnology, and um I was looking into this, a couple of weeks back and hearing about it, but there's so many aspects of technology now that are being applied to, you know, the medicine in general, but more specifically when it comes to, I guess, modifying and attacking certain genes.
Yeah.
Perhaps not attacking is not the word, but like
Targeting,
modifying, I'd say.
targeting.
Targeting, yeah,
Yes.
targeting certain genes to, before, and it goes back to what you research, like before a human is even born, modifying these genes and
Right.
targeting certain genes to then be able to, I guess, make certain, like some genes predominant, and
Yeah.
eliminate some that could potentially be problems in the future, and things like that. I'm not sure as for these specifics. I'm sure I'm probably messing something up,
No,
but
no, this is
what when it comes to your particular research in brain development, what kind of technologies are present now that, or that are people are investigating when it comes to these things?
so. Yeah, so our lab is actually a lab for that makes mice that are genetically modified. We actually target genes, different uh, molecular factors that can actually make us visualize neurons that will be produced from that particular cell that expresses that molecule. And also, in general, the field uses a lot of mutations. So, mutating a particular factor that can affect certain aspects of brain development. One major breakthrough in technology that has happened in the past decade is actually single cell sequencing. So basically, an individual cell can be sequenced for its entire genome, but not just the genome, but the messenger it also. So each cell can actually, the DNA from each cell can actually transcribe a message, which is in the form of what is known as RNA. And with
Right.
COVID, you must have heard of a lot of RNA, right?
Yeah.
So this messenger RNA that is transcribed, the entire messenger RNA pool inside a cell can be sequenced, which means you know the exact information of each of these RNAs. And that can tell us about what fate a particular progenitor can acquire eventually. So a lot of labs have started doing this single cell RNA sequencing technology, and now there are multiple versions of it. And if you are more interested in it, we can go more in depth of that. But basically the idea is that you can target a particular gene, okay, that is expressed in one particular progenitor. And when you start sequencing that progenitor, you know a lot more about what that progenitor may become by just sequencing the different messages that come from it. And that's what the technology is getting into.
Right. Okay.
So
Yeah.
a lot of research now is based on trying to understand, doing these high throughput screenings where you can actually get information instead of doing staining of one tissue, one at a time, one cell at a time. You can actually scan multiple genes in multiple cells inside multiple tissues at the same time. And so I think this is really being revolutionary in the field of developmental neuroscience per se. And I'm actually trying to learn it right now from some of my colleagues so,
Yeah.
so that I can carry it out further. Yeah.
Yeah. I mean, I think it's something that's very interesting, so we can definitely go more in depth. But
Yeah.
I guess one question I have is, what things in particular are you in your lab, or perhaps that you've researched or heard about in other labs, what kinds of things are they doing in terms of gene modification? Like, what are they looking for? What are they trying to change? What are some of the things that they're maybe trying differently or trying out?
Right. Right. So we can target genes at multiple levels. One is at the level of what is known as transcription factors. Transcription factors are sort of molecular selectors. They give a cell an identity. They tell that this cell will become this particular. For example, something that will help my hand move, something in the brain that will coordinate my hand function may be expressing only a certain set of transcription factors, a group of transcription factors, say from A to Z. Okay, so these are 26 transcription factors which will control the movement. So what different labs do is they target these different genes and identify which of these transcription factors is the master regulator, right? And which of them come subordinate to that. Same way, this can be done at a level of morphogens, which are basically molecules that are released into the tissue. And as they are released, the different cells that can see the concentration of this morphogen can detect it at different level and start a different gene program under that. So downstream of that, basically. So different labs can target these different molecules as well that are being released by a particular cell and then identified by another cell or detected by another cell. There are people who do research on neurotransmitters, which basically help communicate between different nerve cells. And not just at the level of genetics, but people have started now working at an epigenetic level, which means how environment can affect different states of a cell or different cell types. And so I think in our lab, we are basically targeting all these different genes as well as environmental components and trying to understand how that can affect development of the brain.
What kinds of what is the goal? What are some of the goals that people want to achieve with this gene modification? I'm sure, for example, a goal would be to be able to detect a baby that's going to be born with a certain disease, right?
Absolutely.
Okay. And then try to eliminate that particular faulty, I guess you'd say, DNA sequence molecule. I'm not sure what it would be.
Yes. So I think there are two kinds of research. One is known as the more fundamental or basic research that tries to understand how a process exists and how, if we perturb it, what will happen. And then there is a second level of research, which is known as applied research, which is what clinicians do, right? They try to get all the information that the fundamental researchers have provided them and see how this can be put into practice. So how can we affect, so how can different genes that affect autism spectrum disorders, for example, or schizophrenia, do we have any idea about that and how they affect brain development? So what you said is exactly what the eventual goal is, to understand how these modifiers can actually be targeted to make sure that no embryo is dyslexic, or they don't have learning disabilities, or they have issues with epilepsy, or they have schizophrenic, and so on. So there are lots of brain developmental disorders that can affect the function of an organism or a human. So the ultimate goal is that more research is now happening on humans themselves, because people have been able to now grow something called an organoid in a dish as a 3D structure, where it is the original tissue is human, and they're able to propagate it inside a dish and try doing these different mutations, looking at these different cells inside a dish and trying to find what is the difference we observe between a human tissue versus tissues that are present in different animals, because that's what we've been using for research, right? So are there differences, and what are the differences in the type of cells that exist, in the type of molecules that exist, and so on. So the ultimate goal is definitely developmental disorders. That's like a very broad goal. And this is what I think hundreds and hundreds of labs are eventually aiming for.
But I also think that it can be used, this technology, I'm sure, can be used for these very proactive and positive things, such as the prevention of disability. But I believe that maybe it could also at some point be used as from an economic standpoint, as a
Mhm.
tool for people maybe in the upper class to then have access to modify the genes of their children, which is kind of like that other side to it.
Yeah, yeah, yeah. No, it's true. The thing is, there is a full field of ethics, like neuroethics and generally scientific ethics, and modification of genes to a certain point will be allowed, and there are lots of rules written down for this, right? Uh, Only when it comes to disorders that are life-threatening or that actually treat disabilities is what should be allowed and will be allowed, and this is what we discuss when we are actually learning about research and learning about science. So yes, there will be people, I agree with you, there will be people who will try to change certain genes, but it's not that trivial. Changing genes or modifying genes, if it was that trivial, it could have been done hundreds of years ago, because we knew what genes, what neurons are, or like 50 years ago we knew what a cell is, and 20 years ago we knew about the entire genome. But because these genes are useful not just in one process but multiple processes in the body, targeting one gene, it will affect different organ systems. So that's why it's not been that easy to understand how it affects the
Right.
overall functioning. So yeah, I agree with you. But there is a rule, there are ethics laid out about how to use this and how to go on targeting. A lot of stem cell therapy has come into place because of that, and you must have heard about it also. So there are
Yeah, I've
lots
heard a
of,
little bit about it. Not much
yeah,
though.
yeah. So there are lots of companies now that are trying to do stem cell therapy, especially for damages in the spinal cord, which help a child in not being paralyzed, or curing certain reading disabilities or learning disabilities and so on.
Right.
So that's been tried, uh, and hopefully it will be successful. But yes, like you said, there are positive impacts, the negative impacts. There are lots of checks and balances that will hopefully not allow that to happen.
Yeah. When it comes to technology, it's not just this aspect of gene modification, but also, it could also be um some sort of development to where, the medical industry along with the technology industry can develop a chip or something that can be
Yeah.
implanted into the brain to then cure some sort of paralysis that someone may have.
Yeah, so actually there is deep brain stimulation is something that clinicians anyway try, which is basically putting an electrode inside the brain and just basically allowing signals to pass through so that it can activate certain neurons. It works in a lot of cases, it doesn't work in some. So I think people are also trying to see which way to go with that kind of a thing. Implanting chips and all is great, and certain researchers also do it, but the brain is a much more complex organ. So I think uh, there are multiple layers to it. So it's not just neurons in the brain. There are astrocytes, that are cells that support neurons. There are blood vessels that can actually provide nutrients to the neurons, and all these cells can get affected by a simple modification. So I think that's something researchers are trying to target, that can you isolate one cell type from the other? Can you isolate
Right.
within the brain these different types or not? And we have been able to. So
Okay. Yeah, there's
yeah,
still, a long road ahead. I think it's only in its,
infancy. Yeah,
infancy, in its initial stages
yeah,
of research, just because just now, recently we've been able to have access to these
that's true.
improvements in technology.
Yeah.
Yeah. Now, as for, the particular research that you do, what kind of things have you been able to do in your lab when it comes to rats? What have you been able to modify, or what kind of developments have you been able to observe?
Yeah. So I think one major advantage of being in this lab is that you can actually follow a neuron from when it's born all the way to when it actually forms connections and becomes a functional neuron. So what we have done in the lab is, I told you about these different transcription factors, right, which help in uh, converting the message of the DNA to the RNA, essentially. And so there are these different transcription factors that are very specific to cortical development in the brain. And cortex is the seat of intelligence. So factors like LHX2 or FESF2 or PAC6, and I'm throwing names at you right now, but just
Yeah.
think of them as A, B, and C, right? So these different transcription factors have different roles to do. And now that we can target them, or we can actually see a fluorescent protein that is within that particular embryonic cell become a particular neuron. So we can actually follow how each cell gives rise to these different neurons, and are they different classes of neurons. So there may be certain neurons that are important to communicate with the other hemisphere. So brain has two hemispheres, right? And there are neurons that can communicate more with the spinal cord, or more like output types. So those kind of neurons, do these transcription factors control the kind of neuron that this particular neuron will become? And if we modify one to the other, will it actually change the fate of that cell? And so these are the kind of tools that we've been using in our lab, and we've been able to acquire a lot of um, lot of research ahead. We have gone ahead and shown that yes, there are fate restricted cells based on transcription factors they express. And so one will produce neurons that project to the other hemisphere, one will produce neurons that will project to the spinal cord. And that itself is a very big deal, because so far people didn't know whether it's like all neurons just come from all these progenitors, but with a molecular handle we can actually tell that this is the difference between the two. um, so that we have been able to do. A different group in the lab can actually also perform functional tasks like behavior, like hand to mouth feeding. Can these different transcription factors control the kind of neurons that will be more important for, say, stretching out your hand versus eating or not? And so I think what our lab has been able to do is to identify certain factors that are important to give rise to neurons that are important for different aspects of their function, not just development. So, and then this could be hopefully translated in humans as well, because same transcription factors exist in humans. So, if there is cerebral palsy, if there is a motor disorder from the cortex or the cerebellum, we can distinguish between the two, hopefully, and eventually get to these different development to function kind of a link, which is missing essentially in the field.
What about, for example, when it comes to the behavior of rats in this case? Is there a way to where in a specific embryo stage, certain neurons can be modified to then have them, behave a certain way once they're born?
Yeah, so I think that's a very cool question. And that's what people are trying to do. So what we do is we don't modify them. We actually just study how in the behavior, like how in development, if we just study this kind of neuron, if we modify them in adulthood, will it give a different kind of behavior or not?
Hmm.
And with that, we do see differences between how different factors can actually change the behavior slightly. Even though it's very slight, you can know that there are these transcription factors that can control a different aspect of the same behavior. And there are many labs, not ours in particular, that have done modifications in the embryo. And if you have to do, because these transcription factors are important for different functions of the body, you have to do it specifically in the brain, specifically in a structure of the brain. And they've done that and seen that there are behavioral differences later on in life. So yes, those experiments have been done.
Right. So perhaps in the future, if two parents tend to have some sort of impulsive, angry behavior, it could then, be looked at the infant that is going to be born at the embryo and then try to modify it to, maybe have a less aggressive trait. Is that towards where this would be perhaps going?
I think I think right now the focus would be more for larger disorders, like where there is
Right.
life-threatening issues or like lifestyle issues like autism or schizophrenia or things like that. Anger management is something I feel people are still trying to get done more with therapy rather than gene targeting,
Okay.
because again, like you brought it up earlier, that it's more like a rich person's thing rather than
Yeah.
getting it done. So that's why I think it's better to first focus on disorders that are actually affecting their presence in the society and affecting their livelihood, rather than, anger management can be done with therapy. You brought a very nice example. So I think that's the idea of gene therapy, right? Like, where people think that they can use it for anything. We actually don't even understand what are the different factors that make a person angry. There are different transcription factors. So like, knowing the genetic makeup of that, the kind of environment they grew up in, how. So there's been a very nice group there are many nice groups that study stress management and stress in so early life. So if early life stress is there, in adulthood, the organisms, whether it's mice or humans, are more prone to anxiety and depression and anger issues. And so a combination of genetic factors as well as how they are being brought up makes a very big difference. And I think those are the kind of therapies people are trying to target for anger management or stress management, rather than trying to modify the genes. But yes, genetic profiling is very important, because now we understand there are certain genes that, if mutated, will cause this issue. So, for choosing your partner or choosing someone, you can actually go through that list and see whether uh, your offspring will have those issues or not.
Right. So the
So
focus,
right now it's more preventive rather than cure.
yeah. Okay. Yeah, the focus tends to be and also more on, and like you said, on these bigger, more prominent issues, you could say.
Yeah, more prominent issues and issues that actually affect the life or death or the quality of somebody's life more
Yeah.
than, yeah.
Okay. That's
Yeah.
interesting.
Yeah.
Do you see this, or I guess what future do you see maybe in your lab, in this, um of course in the future, it will try to gravitate towards then now trying to experiment with human life form instead of, animals or other animals, right? But in your particular lab, for the foreseeable future, how do you see things developing and evolving?
I think one thing, like I said, is not known is whether you can connect development with function at all. Can actually changes in gene structure during embryo, can it actually affect the functioning of a body or not in a particular way? Can you actually target, say, transcription factor A and see behavior A being affected or not? So that's something which is unknown, and that's something that I think it'll be great to explore, and that's what we would try to explore. Um, and secondly, trying to understand at a bigger level how these different genes interplay between each other to give a certain phenotype or a certain outcome, whether it's cellular or behavioral. Behavior is much more difficult because cellular outcomes are much easy to see, observe, whereas behavior is a combination of different factors, like I said, environment as well as genetics. I think that takes much longer. I think that's where, so I think, in short, if I want to just put it in one line, I would like to understand the developmental basis of functioning of a brain.
Okay, I see. Yeah, that's interesting. Yeah, and it has a, I'm sure it'll keep progressing in a lot of different
Yeah.
ways. Something that comes to my mind right now, as you were saying this, and I was thinking, is I feel like at some points during the development of the brain in an embryo, you can't really modify things anymore. As for the stages of development
Yeah.
within, a womb, or within a mother, um, or in a, I guess, because sometimes it doesn't even have to be in a, like I think it can be like in an artificial womb now. I'm not sure, but
Yeah,
I'm
no,
not sure what
it
stage of
starts with
technology.
artificial, but then it has to be implanted in a real one.
Okay. Well,
Yes.
so really, so at what stages of development of an embryo, can you modify the brain, and
Yeah.
or certain genes within the brain and molecules, and also at what stage does the brain start to form, and kind of the different parts of the brain?
Yeah, that's a nice and broad question, and basically covers the entire field of development. Um, so in humans, I will probably talk about humans. The development of the entire brain starts around gestational week 5 or 6. Um, and that's when, the embryo has 3 different layers. One of the layers becomes the skin and the central nervous system, which includes the brain and the spinal cord. So this is known as the neural plate stage, which is basically when the embryo knows that this part will become the brain. Okay. And then this plate basically folds into a tube. Once the tube is formed, there are cells that line this tube, which are the mother cells, which are the progenitors. And then they get specified to form different kinds of neurons. Now, this specification happens at different times for different parts of the brain. So the first is the spinal cord, which happens around gestational week 7. Then happens the hindbrain area, which controls, more reflexive behaviors like respiration and so on. That is the next one, which is why when you look at a baby, when you look at an embryo, you will start seeing random movements like the diaphragm is moving or the chest is moving. And that's more reflexive behavior, even though the embryo is not really breathing at that point, but it's trying to learn that, okay, this is when it can start breathing, this is how it has to breathe. So that is known as the fate specification stage. And the last one is the cortex, which is the seat of intelligence. And that happens around gestational week 10 or so, 10 to 15. And so during that phase, which is fate specification, is when progenitors decide that this is the neuron we are going to give rise to, or we're going to produce. Okay. Now, once the neuron is produced, there are more fate specification happening within those neurons to become neuron A versus B versus C. So the progenitor has given rise to neuron ABC. Okay, and then within the neuron, it will decide whether it has to become A or B or C. So fate specification or proliferation is the stage when you can change genes, you can alter the fate of a particular cell and the behavioral outcome. And that is when most of the changes happen.
What week was this again? What gestational week?
This was between, so you can start from week 5 through week 14. This
Okay.
is the entire period when the brain is being formed. And these are the modifications that you're doing at the time the neurons are being born, essentially, right? So that's the fate specification type. Then there is something called neuronal migration, which is the next phase of development, where neurons actually, when they're born, they actually migrate out to different sites, right? And that is when you can also affect. So, you know how our brains are very convoluted. There are gyri and sulci on all these formations. There are mutations that can affect migration that will make the brain smooth, just as in case of a rodent. And that can affect cognitive abilities, that can affect learning abilities, and so on. So migration is another point where mutations have been studied that affect brain function. Third is once the neurons settle down where they should settle down, and this is when the baby is actually being born. This is perinatal. So this is when it's being born. Neurons decide to produce axons, which are basically processes that connect to other neurons, to communicate with them, to decide what they need to do. And that's just after the baby's born is when this process exists and happens. And that is the third phase where you can affect the genes as well. And here the environmental component becomes much more severe. Like what is the kind of environment the baby's being brought up in and so on.
Do you mean that's before it's born or once it is born?
No, right when it's born.
Like okay.
Right after it's born. Before it's born also gets affected, but it's more affected by what the mother is ingesting rather than directly affecting the baby. So, if there is stress around the baby, it'll definitely affect the brain development, because that's when the axons, the neurons are trying to communicate with each other. There are lots of neurons that were born are actually being killed off because they're of no use. So that process is known as pruning, and the correct synapses are being formed. Correct synapses as in two neurons communicate with each other, and that gets strengthened if that is the correct one. And the other ones get weakened and gets removed, the ones that are not right. And so this is the fourth stage where it can get affected. So there are these multiple stages where mutations can be done and can affect how the brain will develop completely.
So you can still do a mutation once the baby has been born?
Yeah.
Because how would that be?
So there are certain genes that, in fact, there are many genes that can affect the migration, can also affect axon pathfinding. So basically how the axon finds the other partner. When a neuron tries to find the other partner, you can actually create an issue even then. So nobody mutates a cell at that particular time. But in mice, we can always do that with injecting a virus with certain mutation into that gene. So it gets integrated and it can affect the development of that particular neuron. And that's how you can study how even after the animal is being born, or maybe like the first week of its being born in a mouse, or like which is equivalent to like the first 5 years of human, you can actually have learning disabilities, language disabilities, visual disabilities, and so on.
Okay.
Yeah.
So that those first days, weeks of a baby being born are extremely important as for their development in the future.
In fact, in the human, the first 8 years of their life are very important for
Yeah,
them.
yeah. In childhood and so on, I'm sure. But
Yes.
is there a specific importance to those first couple weeks? Or is
So,
it just equally important, you would say?
no, I would say there is a difference because there are different
Okay.
things that are happening in each of those. The first couple of weeks is when the neuron is trying to find its partner. The next months or so, or a few years, couple of years, is when there is a lot of pruning, which means a lot of wrong connections are being removed and the right connections are being strengthened. And then for the next part, which is known as critical period or sensitive period, is when different sensory systems get formed differently. So there'll be a different time, a critical period window that will be important for, say, taste. Then there'll be a different one for vision. There'll be a different one for hearing, and so on. So I think each of these, actually, visual system develops till very late, till almost like 7 years or 5 to 7 years of age in a human baby. So I think all of these factors, environmental factors in particular, can affect this phase of development. And I think a development
Okay.
is considered to be done only after that point.
Right. I mean, your brain doesn't fully stop developing, I believe, until you're 25 years old, right?
Well, that's not true technically.
Okay.
Yeah, but there is a lot of plastic changes. Like once it's formed, there are new connections being formed, and that keep happening in certain regions even up till the person is old, which especially in the hippocampus, which is important for you to recognize space, understand navigation skills, and so on.
Okay.
So yeah, but that's not considered as early development.
Right. Okay.
Yeah, yeah.
Now, I think it's important to understand what exactly can affect development. As you mentioned stress, for example. You
Yeah,
mentioned, alcohol use, I'm sure drug use,
yeah,
um,
definitely.
poor health, all those things. So what
There
can
is.
affect the baby, and like what can a mother do to prevent or to ensure that the best possible health for for the embryonic development?
Yeah. So I think there are many different factors. One of them, which I will talk about, is definitely COVID or viral infections, influenza. Different infections in the mom can actually affect the development of the brain of the embryo, of the fetus. And there has been a study in 2021 itself that has shown that it actually affects the cognitive ability or learning ability inside the embryo if the mother catches COVID infection. So that is why vaccination is very important, especially in pregnancy. So that definitely, influenza has been studied at length for like how flu can affect the baby if the mother gets the infection, especially in the third trimester. Yeah. And so that definitely, so infections is one. Second is proper nutrition. And by proper, I mean not to overdo it. So there was a recent study in mice, it's not been translated to humans yet, but that showed that if you give overdose of folic acid to the mother, it can actually affect the development of the babies. So, so like just the appropriate amount that is being prescribed by the doctor is what should be taken at that time.
What does folic acid do in the pregnancy?
So it's vitamin B, it's
Okay.
vitamin B9, and it's important for multiple ways of like, especially as an antioxidant, different oxidation stresses that exist inside. In fact, alcohol is also similarly damaging because it causes oxidative stress. Um, and that's what causes different tissues to die at different levels. Narcotic drugs, you should definitely avoid opiates and those kind of drugs because there have been differences in the dose that have been observed and how it affects the brain development. So at smaller doses, it affects the kind of cells that are being born, but at larger doses, it'll cause the entire brain to shrink. It causes that kind of a drastic difference. What else can I think of?
I may
Stress,
be wrong.
definitely.
Just something that I have in mind with the drugs. I may be wrong, but
Yeah.
you can answer this. When let's say a mother were to take some sort of drug during her pregnancy, not medical drug, but like a recreational drug or whatever, would the baby perhaps then, or the embryo, would they perhaps then have this once, because they're developing, would they maybe have this like altered sense of reality just because that's what they lived once they were in the womb?
No, so
Okay.
that's not what happens. It's not
Okay.
an altered sense of reality. It's just, it comes out as, disabilities to learn, or, things like having epileptic attacks and these kind of things are what happens. It's not that because that drug creates such an effect in the mom, it's not the same effect that the embryo will feel once it's a baby, that's not the case.
Well, no, no, not once a baby. I meant like perhaps like it would just affect development in that way. But I was you explained it better. It creates disabilities probably because it's not the normal way for the brain and the neurons to develop, and the mom isn't can't provide those natural, or like it's not in their in her normal senses. I understand what you're saying. Yeah, okay. So I had a wrong conception of it.
Yeah, it's not like it'll affect directly the baby. It's that because the drugs have actually reached the embryo, there are different ways it does when the embryo is being formed and when the brain is being developed. And so it'll affect processes that I just talked about, which are phase specification, migration. And so that affecting of migration can actually cause the baby to be born with a smooth brain instead of a more gyrencephalic brain,
Yeah. Okay. I guess you explained it. You explained it well there. I guess my question was whether the baby absorbs a substance that's making
Yes.
this drug the drug, and now you're saying that it doesn't. It's just
Like it can absorb, but it will not have the same effect in a baby
okay.
as it haven't. Yeah.
Got it. It's
It
more so
depends on
in
the
the
drug.
developmental. It's more so in the developmental part of the
Correct.
brain and affecting how the brain will develop.
Correct. Exactly. Yeah. Not the
Okay.
functioning. Yes. Functioning will be completely altered in a different way. Yeah.
Got it. Okay.
Yeah. But that's a
Uh,
very cool question. I think that was very nice one.
yeah, I just wasn't sure.
Yeah. Yeah.
And what can a mother do to ensure that the baby is born with optimal health?
Well, take care of herself. And I think more than the mother, I think the partner, the spouse should be the one making sure that the mother is also in perfect health. Give her as much care and love and nutritional food to eat, and also keep her happy, because I think happiness of the mom definitely affects the child. These are all anecdotal evidence so far in different cultures. I'm sure even in Mexico you must have heard something similar, but there's scientific evidence to, if the mother is stressed, how it affects the embryo. In fact, when there are infections, which is why it affects embryonic development, is not that the infection can penetrate through the embryo, but infection creates inflammatory responses in the mother, which then affects the development of the embryo. So I think just making sure that it's a healthy, happy environment for the mom can actually lead to a healthy, happy baby, is something
Right.
that we've heard everywhere, but there is evidence for it too.
Yeah, no, I'm sure it makes sense. It makes sense that if the mother is happy that the and she's well and she's healthy,
Yeah, no
it's
alcohol, no
yeah.
recreational drugs, no nicotine, none of those.
Yeah, it makes sense. It's just like, I've seen or I've heard about studies where, where the music is played for the embryo while it's still in the mother's womb, and how
Yeah,
this could potentially help. But
yeah,
you don't think about it necessarily as a medical, um I guess, approach, but it's still, it's these external things that
right.
make sense that they could still in some way or another could help the development of the
Yeah, so
embryo.
there is research that is being carried out. There are a few labs that actually do this, like, how music therapy can affect cognition and so on. But because the research is not completely in one direction and people don't really have a solid answer, that's why scientists are not able to tell the audience or tell the general public that, this is definitely going to work. That doesn't happen. So I think that's the thing. But yes, definitely what you're saying, playing happy music or something like that can definitely affect the abilities of an embryo, of a baby rather, after it.
Right. Is there should a would you say that there's a difference in a way a mother should approach pregnancy in the different trimesters or in the different phases of pregnancy?
Oh,
Like,
definitely. So I
okay,
am not an expert at this, but I will definitely tell you that first trimester is very different from the second, is very different from the third, because like I said earlier, there are different events, developmental events happening at three different stages. And so I think the first stage is the most important, because that's when the specification is happening. And if that goes wrong, the baby can even die, it can be that bad. So
just a quick then you could continue, sorry, but when do you know major diseases you mentioned, for example, schizophrenia or autism, when do these develop?
so it can happen at multiple points. It can happen at the stage of specification, which is as early as 5 weeks,
Okay.
to say during migration, which is when neurons can't migrate properly, and that can lead to these disorders, or even till birth, like at the time of, like, very close to birth, when the axons are being formed. So I think
Okay.
these are, like, it can affect schizophrenia and autism because they are multi-mutational diseases. There are multiple things that can cause it. It can happen anytime across these three trimesters.
And probably complications at birth as well, such as, lack of oxygen and
Absolutely.
things like that. Okay.
Yeah, yeah.
Okay, well, you can continue talking about the different trimesters. That would be great. Sorry for interrupting. I was
Oh, no, no,
curious.
no, no worries. I was basically saying that these three trimesters have very different events happening in during the child's development. And, a lot of this is affected. It's not just the brain, right, that's developing. There are different organ systems that are being formed at the same time. So the first trimester is considered to be very crucial because of that, because it's not just the brain, but the blood, the bone, everything is being formed at that time at a very large, like, very fast pace. And especially during the brain, so the between the first and the second trimesters is when the brain development is most crucial in terms of making that particular neuron or making that particular astrocyte or so on. So I think, well, the doctors are much better at this to tell you which of these trimesters are important, but as a developmental neuroscientist, I would say that first trimester is the most important to even specify that this will become a neuron. The second and the third become more important as to whether the brain will be smooth or convoluted or not. And the third one will be proper functioning. So I think any of these phases will affect, but in different ways.
Okay.
So
So really the health of a mother and of the embryo development should be prioritized throughout the three trimesters, and it should it be approached differently, or is more or less the same?
I don't know the answer to that.
Okay. Yeah,
And I
okay.
think it's a question for a more specialized medical doctor rather, I would say. Uh,
Okay. No,
yeah,
I was curious. It just,
yeah.
I'm sure that the health
And there
is
are different
important
ways.
throughout the whole time.
I would say there are definitely different ways to deal with the three different trimesters. And the third trimester in particular is more to do with how big the fetus becomes, right? And the mother has to deal with different kinds of issues then too, because the fetus is so big, she has to also take more care about her balance, her back, and so on. Earlier on, say in the first trimester or up to 4 or 5 months, it's more of a physiological change, but after that it's more of the appearance as well. So I think, yeah, the
Okay.
physiological changes happen earlier, and that's when most mothers have issues, which is why it has to be, most doctors advise that the first half of pregnancy is more crucial. And yeah,
So that first trimester, it's what, when it's starting to, I from my understanding, it's when it develops the cortex, right?
it
And
develops
and
different
this
kinds of
cognition phase.
spinal cord. Yeah, spinal cord. Cognition
Okay.
phase comes much later.
Oh,
It's
okay. Okay.
when it develops the spinal cord, the hindbrain, which helps in breathing and things like that, more reflexive actions, and then the cortex. So I think between, say, 5 weeks to 14 weeks of gestation is when it is very important that these things are becoming what they should become in the first place.
Right.
Then there is migration and they'll reach where they should be reaching. And so, yeah, so I think the first 2 trimesters are important for these processes.
Okay. That's interesting. There's a lot of information to
Yeah, it's a lot of
digest.
information. Yeah, which is why I was like, I don't know how to give you one line answer for this, but there's lots happening during that time. And different events are happening at that time. So, if there is a mutation which affects it, at any of these stages, it will affect brain development. So,
Yeah. Well, I think we covered a lot of things.
yeah, we did. And I hope I didn't confuse you with lots of terms. But
No. It's whenever you learn a term or something specific for a certain field of study or something, it's important to explain along with it, and you did a good job of that. So
okay,
whenever
I'm
you
glad.
mentioned
I'm glad.
something, you explained it, and so I understood. Yeah.
Great.
I'm not gonna remember every term you told me, I'll tell you that, but I will remember like the
No,
explanations.
totally. If you have any questions, you can also email me or anything, or just text me now that we have each other's phone numbers. Yeah.
Yes. Well, what you do is very interesting, and it has a very, for the future, it's very applicable, just because this gene modification and these gene progression and this embryonic stage. I mean, it's something that it's very, prominent in medicine. So it's interesting, and thank you for sharing.
Thank you. Thank you for giving me the opportunity. It's been great talking to you.
All right. Well, take care.
All
Thank
right.
you very
Yeah.
much.